This is a production of Cornell University.
Thanks. It's really fun to be here. As Bill said, I did my undergrad here, and there were a couple of faculty members here when I was an undergrad, specifically Harlan Banks and Dominic Paolillo, that had absolutely profound impacts on my entire career. And by extension, they actually impacted Duke students for the last 25 years, because I thought of those two Cornell faculty whenever I encountered an obsessive plant person, and I really wanted to help them out when they were undergrads. So I have warm feelings for Cornell, so it's great to be here. So Sphagnum is one of the larger genera of mosses. It's also an entire ecosystem that forms really a subfield of ecology, peatland ecology. So it's really kind of unique. It's really kind of unique in that way. Peatlands are found throughout the world. They're not all sphagnum-dominated peatlands, especially down in the tropics. But that area in the north, up here, I think that shows, doesn't it? No, it does not. That area up in the north, these are mostly almost completely sphagnum-dominated peatlands, and they're very important. They only cover about 10% of the land surface of the Earth, but they account for 25 to 30% of the entire accumulated pool of terrestrial carbon. So they're pretty remarkable. And they occur where there's an unbalance between growth and decomposition. So growth can be slow, but if decomposition is even slower, then peat will accumulate. And peat, to an ecologist, has a definition. Some X number of centimeters of accumulated peat defines it to a vegetation ecologist as a peatland. And they come in all different shapes and forms, and they're almost all absolutely beautiful habitats to spend time in. This is a typical boreal peatland. They may or may not have open water, and where I'm standing out here would be a floating mat on that open water, really dominated by the sphagnum mosses. But they occur, as I said, all different shapes and sizes. This is a peatland in southeastern Alaska, where it's basically rainforest kind of ecology. And here's a peatland in North Carolina, where I'm coming from, and this is a site that we've gone to many times for research purposes, surrounded by bald cypress. And these peatlands in North Carolina also happen to be the home of the famous Venus frog trap. I was surprised to see one time when I was there. I figure any plant that is gobbling vertebrates is one to be a little bit careful of out in the field. But carbon is stored in peatlands. They're said to be a net accumulator of carbon these days, possibly about to change to be a net source of carbon into the atmosphere as climate warms. But for now, and for the last 10 to 20,000 years, they've been accumulating carbon, and that's in the form of peat. So this is a core through a peatland, and the sphagnum is up there at the top, but it accumulates this large amounts of peat, and it's basically almost pure sphagnum. And there's a number of reasons why peat accumulates in these kinds of ecosystems. Some of them are extrinsic. These are waterlogged and anoxic kinds of things. They're usually in the boreal zone, cold climate. But there also are intrinsic features that are characteristic of sphagnum, a variety of chemical features that lead to very low decomposition rates. So you can transplant a sphagnum into a non-cold, non-anoxic environment, and they still decompose slowly. So exceptionally slowly compared to any vascular plant. So the stars, if you will, of these peatlands is the genus Sphagnum. And Sphagnum is a moss. And they have the very characteristic bough plan that makes Sphagnum, to my eye at least, relatively easy to recognize compared to any other moss. They have this capitulum at the top of a cluster of branches. And the life cycle is a typical moss life cycle. So the free-living plant that one encounters out in nature is the gametophyte, typically haploid, but they're spore-producing. So when the spore germinates first, there's an immature protonematal stage, and then that gives rise to the mature gametophyte. Depending on the species, the gametophytes may be unisexual, and different gametophytes have archegonia, female, or Antheridia male, or there are a minority of species that are bisexual gametophytes that have both Archegony and Antheridia on the same. Fertilization requires water for the sperm to reach the egg, which impacts gamete dispersal distances and therefore gene flow distances. But the result is a sporophyte, and the sporophyte is very small, basically nothing but a sporangium, that black part up on the top. Everything about sphagnum is all about water storage and control of water movement. So this is the structure of a sphagnum plant with that capitulum at the top with fascicles of branches. They have dimorphic leaf cells with large, empty, dead at maturity hyaline cells and enclosed in a network of these green photosynthetic cells. And those hyaline cells are absolutely chock full of microorganisms. And I work with a lot of, I'm not an ecologist, but I work with a lot of people that are primarily interested in the microorganisms associated with the sphagnum and relationships between the microorganisms and the host sphagnum plant. And it is a symbiotic relationship, not just an association. So sphagnum, it's the phylum or division Bryophyta, the mosses. It forms a class unto itself. I'm not going to have time to go into it today, but we've worked on the deeper level phylogenetics of the Sphagnopsida, and I will argue that there are 3 genera of Sphagnopsida, 2 of them very small, and then Sphagnum, actually that's wrong, 4 genera of Sphagnopsida, 3 of them very small, and then the genus Sphagnum includes up 150 to 400 species. The diversity of them is not very well understood, which by moss standards is actually quite a large genus, maybe not by legume or grass standards or something like that, but it's a large group. And they have they're distinct from other mosses in virtually every characteristic of both the gametophyte generation and the sporophyte generation, including these dimorphic leaf cells. They typically have no rhizoids. Mosses never have roots, but they're usually attached to the ground by these filamentous rhizoids. Sphagnum has no rhizoids except 1 species outside the genus Sphagnum that has been reported with rhizoids. And the sporophyte generation is unique compared to any other moss as well. Now, for well over 50 to 75 years, sphagnum-dominated peatlands have been a model for community ecology. Oh, can you do that? Oh, I see an X there. Boy, great mind. Thank you. Is that what that X means? Sphagnum-dominated peatlands have been a focus for community ecology, basically because they're relatively simple communities. They're boreal communities. We're not talking tropics here. But especially species sort themselves along niche space very finely. So they've been a model for community assembly, community structure. And these figures show a couple of examples of that. The species sort very regularly with regard to hydrological gradients along this one, from the margin of a peatland to the center. This figure is accumulated data from 50 years of peatland ecology, showing how species sort themselves out relative to cations especially. And then in the bottom right, a student of mine was studying pH variation in terms of the niche of these species and superimposing that on a phylogenetic hypothesis, and demonstrated that not only are species distinct, but the similarities and differences are phylogenetically conserved. That it even runs in deep phylogenetic trades within the genus Sphagnum. Now, Now, hydrologically, they've been very interesting for peatland ecology. They sort, as I said, along these hydrological gradients, and you can see a species here and a species there. And the change in elevation above the water level is measured in a centimeter or 2. So they're very sensitive, and you can see how sharp that is. Some individual species are very characteristic of low so-called hollows within peatlands. So this is Sphagnum cuspidatum. Other species at the other end of the spectrum form hummocks. And this has been a focus of peatland ecology for many years. And the interesting thing about this hummock is that you can see that this is strong enough and big enough for my collaborator to be climbing across it. There's no topological variation under that hummock. It's the sphagnum that builds the hummock. In other words, there's no ground change in topography. So that hummock hollow divergence between species has gained a lot of attention in many years. And at one end of the spectrum, there are these species that occupy hollows, and they tend to have high decomposability within the context of sphagnum, high growth rates, but they don't form hummocks because they decompose too quickly and they don't build up peat. At the other end of the spectrum, there's species that form hummocks. They grow slowly, but they decompose exceptionally slowly, so they build up these hummocks. And we did a meta-analysis, a former grad student of mine, and a number of metabolic and biochemical kinds of traits. And you can see these are correlated traits, suggesting that there's a whole adaptive syndrome, if you will, that underlies this hummock hollow topology, microtopology within sphagnum. Arguably, one of the most interesting traits to look at is decomposability per se. Kind of an interesting trait if you look at it, if you think about it, it's a part of the extended phenotype. It's a phenotype of these plants that you measure after the plant is dead. It's the rate at which it decomposes. And my student, Brian Pietkowski, did quite a large decomposition experiment. As it happens, he did it at McLean Bog, belongs to Cornell, right up the road here. And he planted out bags of a whole suite of different species of moss, let them decompose for 3 years, and this is a standard approach in peatland ecology, and measured variation among species, and also superimposed it on a phylogenetic hypothesis, and found that decomposability is not only species specific once again, but is phylogenetically conserved. We know from 50 years of peatland ecology, we know how these species sort relative to height above the water table within peatlands, that is hummock height. And we know that there's a correlation. And here the colors correspond to these clades. So there's a clade that tends to be high decomposition, low, these are hollow species, these are hummock species. And then if we take the next step and superimpose all of this, we see that shifts in niche phylogenetically are correlated with shifts in decomposition rates, incredibly sensitive to changes in niche. I accused my then graduate student of making up the data, kiddingly, because the correlation was so remarkable. So all of these features of sphagnum led a collaborator of mine, Dave Weston at Oak Ridge National Lab, to propose to the Joint Genome Institute a Sphagnum Genome Project. And we did get that funded, and it created reference genomes for a couple of species, Sphagnum and magellanicum, and it'll become apparent in a few minutes why I put that in quotes, and Sphagnum angustifolium, and then resequencing a panel of a so-called diversity panel of other species and other accessions. Using that as leverage, we got a 5-year biodiversity dimensions of biodiversity grant, and we resequenced a whole slew of additional accessions representing the Sphagnum magellanicum, which turns out to be a complex. And we also did a lot of RAD-seq analyses, because that allowed us to work with dried material and increase our sample size, et cetera. And we also, Dave's lab especially worked on sphagnum microbe interactions, and we've had funding from the DOE for that work. And so my two collaborators most closely are Dave at Oak Ridge, and Adam Healy is a computational biologist who really did the heavy lifting in terms of dealing with the genome data. So I don't want to spend my time talking about the genome project per se, because from my point of view, that's just background information that facilitates what we want to do in terms of learning about evolution. But so I'll just tell you, there's a high level of synchrony between these two reference quality genomes. The average recombination rate was quite interesting. It turned out to be quite high, about the same as the moss model Physcomitrium patens, about a third or 2/3 higher than the model liverwort Marcantia. And compared to most vascular plants, the recombination rate in these sphagnums are an order of magnitude higher. And that has a number of ramifications. One is it can facilitate adaptive evolution. by a lot of recombination. It can facilitate the purging of deleterious alleles. And so it's a useful characteristic. So more or less the only, well, one of two only other things that I'll tell you about the Genome Project is that we were, through synteny studies and also studies of genome duplication, we were able to reconstruct the genome of Sphagnum from 5 original chromosomes with a duplication and some translocations, and to the current number, which is quite uniform across Sphagnum, 19 chromosomes. The other thing that we discovered with this Genome Project was a previously unknown, very small sex chromosome, chromosome 20, and through synteny comparisons, we were able to demonstrate that that sex chromosome, which is chromosome 20, is a modified version or reduced version of chromosome 7. So we were able to pursue that. Now, we did a bunch of work on the sex chromosome and its biological significance, et cetera, which again, I'm not going to spend a lot of time talking about, but probably the most interesting result was that we detected a QTL from doing single spore isolates and growing them up that impacted the growth of Sphagnum under pH stress. And as I say down here, there's we identified a chromosome, we identified a locus on chromosome 10 that in males is detrimental at neutral pHs, shown there, but under pH stress, either very low pH or high pH, it actually facilitated it. It reduced the impacts of stress. So this was a really interesting example of being able to link this QTL to a trait that impacts where Sphagnum grows within the peatlands, but also impacts carbon accumulation. So it's, in a way, it's going from this genetic interaction to global scale biogeochemistry. It's just a start, but it's a good start. From my standpoint, identifying the sex chromosome was most valuable because it facilitated downstream studies of population genetics, population genomics, evolution. I find the sex chromosome per se extremely interesting, but what it allowed for us as more kind of field people and interested in natural populations was that it gave us markers for identifying male and female gametophytes, whether or not they were actually expressing gametangia, the Archegony or Antheridium. And since they're more often than not expressing they're more often not expressing gametangia, this was a really valuable tool. So we actually developed 2 different tools. We developed primers from the U and the V, the sex chromosome, male and female, female and male respectively, the U and the V sex chromosomes that could distinguish, and we made sure that they worked by doing this with plants that we actually had sexual expression, so we knew what sex they were, and found that they worked reliably. We've been doing more work with RADseq, and we actually developed a method for distinguishing male and female gametophytes using RADseq data and I'm not going to go into the details, but by comparing coverage to the coverage of a typical autosome, because the reference genome we know was one sex or the other, and so we were able to make that inference, and we linked inferences from this to inferences from here, and these inferences were grounded in known sexuality, so we know that these work. And again, I'd love to talk to you about sex ratio biases and clonality and all sorts of things, but I don't have time today. But among other things, it allowed us, for example, we have one site where we have very good microbial data, microbes that are associated with the sphagnum. And interestingly enough, we found that male gametophytes at that site had much lower diversity of microbes associated with them than females. And I'm intrigued by that because this is a species that has a female sex bias, both in that population and elsewhere. And it raises some interesting questions about the microbes, maybe what's underlying that sex ratio bias. But what I want to spend the rest of the time talking about is my favorite subject these days. It's this species called Sphagnum magellanicum. Sphagnum magellanicum is shown a plot of just GBIF plotted distribution found throughout the world. It was described in the 1800s, 1879, I believe, from Tierra del Fuego. but it's kind of a morphologically very distinctive looking thing. It's the only member of its subgenus that's red. And so there were similar looking plants in Europe and North America, so they all became known as Sphagnum magellanicum. And they've been known as Sphagnum magellanicum from 1879 until about 3 years ago. And it was great because even peatland ecologists could identify Sphagnum magellanicum. You didn't have to be a Sphagnum taxonomist or anything. So there's hundreds of papers that deal with Sphagnum magellanicum in the field of peatland ecology. However, the bad news started when my collaborators in Norway got a sense that there were two different things at a peatland in central Norway. And they used, and I think this was RAD-seq markers as well. And most of you are probably familiar with the program, the software Structure. But if you're not, each one of these circles is an individual, and it identifies different genetic groups, not clones, but genetic groups. So suffice it to say for that. And at the site in Norway, they found that there were two isolated groups. There were two completely distinct genetic groups in what we would have called Sphagnum magellanicum. They extended that a bit to a more global sampling and found that, the two groups were still distinct, but intermediate individuals there were individuals that were intermediate in that they were genetically admixed between those two groups. And they went ahead and described these two groups as Sphagnum medium, and which a name already existed in Europe, and Sphagnum divinum, which we described as a new species. And Sphagnum magellanicum per se is limited to Tierra del Fuego from where it was described, because that turned out to be genetically distinct from both of them. That's genetic similarity, that's structure. It's a phonetic kind of approach. It's just genetic differences. It doesn't tell you anything necessarily phylogenetic. So they did, with the same data, analyze it in a more of an explicitly phylogenetic context, and found that sure enough, Sphagnum medium and Sphagnum divinum represent two clades, very clear clades. And furthermore, they argued, or they documented to some extent, that Sphagnum divinum grows typically most commonly near the margins of peatlands and even in the forests surrounding the peatlands, whereas Sphagnum medium grows out in the open peatlands. And so they recognized that there was some kind of ecological differences between them. Well, that got us going because we were in the middle of this Genome project and our Dimensions of Biodiversity project, both of them focused on Sphagnum magellanicum. And we thought, we're in trouble. So we undertook a study and we increased the sample and sampled from Tierra del Fuego, but also elsewhere from South America, and intensive sampling in North America, both in the East and the West, and in Europe and in Asia, scattered around a little bit. I should say that our sample size, since we put together this slide, is at least doubled, if not tripled, but the story hasn't changed. And these are my collaborators on this work. Marta is a postdoc, and Blanca, who is a staff person in the herbarium at Duke, and my former student, Brian Pietkowski, who was at the time the Oak Ridge National Lab. We had a lot of genome resequencing data. And so looking at that first, what we found was that there are indeed Sphagnum medium distinct. There are no support values on there because, as is typical with genome data, they were all 100%. You can get 100% support even if it's the wrong answer. We know that from genome data. But I don't bother to put support values on there. Sphagnum medium is there, Sphagnum divinum, which are the two European species. But then we detected two additional things in North America that hadn't been part of the European study that seemed to be something new yet again. And then here's the South American Sphagnum magellanicum, which we corroborate was distinct from all of these things. Oh, and well, I should just point out, I want to go into it. This is pulling out pretty much whole chloroplast genomes. And there's some mixing of chloroplast genomes, but a lot of the mixing is within these individual species, though not all of it. But I'll leave that. That's a different story once again. So we expanded from that genome resequencing data set with RAD-seq and increased the sample size. And this absolutely invisible tree was the data at the time, but here's a summary of it that you actually can see. And we had got very well supported relationships, and two different ways of analyzing it, and they only differ in one little detail. So what we see, well, I'm going to focus on the North American species. So I've summarized the relationships among the North American species here, medium and divinum, the two that are also found in Europe. But then these two new things, Sphagnum magni and Sphagnum diabolicum, that are very closely related sister species that diverged most recently. And then there were a couple other things that we did resolve that don't have names, which is why I have kind of informal names that are not in italics. Those haven't been described, or we haven't found an appropriate name for them yet. This just shows that the RAD-seq data, while they're just little snippets from all over the genome, they provide good coverage of all 19 chromosomes. Each one of those vertical lines is a RAD-seq locus. So we get good coverage across the genome, and we were able to do analyses at the chromosomal level and look at relationships. Now, I don't know, you probably can't read these numbers, but the punchline is written up there and down here. This is summarizing numbers of fixed differences between those species that we had RAD-seq and genome resequencing data for. And the bottom line is, as you can see from the topology, Sphagnum and medium and divinum differ by thousands in the case of RAD-seq data to millions of fixed nucleotide differences between those species. Whereas these two, which diverged relatively recently, differ by only hundreds in the case of RAD-seq data, or thousands, tens of thousands in terms of genome resequencing. So they really clearly are, those latter two are really clearly much more closely related. And that's shown here. Here's if you just, and this is just kind of a selected plot to illustrate this. Divinum and medium, no question, nothing intermediate, and no question about what represents sort of a biological species. The structure, phylogenetic structure, just turns into a fan when you get into these species because of interbreeding. Whereas diabolicum and magni, what this illustrates is they're clearly distinct from medium, but in this particular case, diabolicum is actually paraphyletic with magni nested within it. And although I didn't have time to go into the details chromosome by chromosome, what we found is that some chromosomes resolve them as a paraphyletic, one nested within, magni nested always within diabolicum. Other chromosomes resolve them as reciprocally monophyletic, but they are very closely related. That's the take-home message. This is what you see if you go out into the field. So these are pictures from one site. They have that magellanicum look, but they vary in color from almost completely green, some individuals really completely green, to just deep, beautiful burgundy red. And they also vary in growth form depending on how high of a hummock they're forming. Higher, denser plants and lower, much looser plants. And so they're incredibly variable in terms of morphology. Now, I like to think that, especially the three of us, me and my two collaborators, Marta and Blanca, are really experienced at going out and looking at sphagnum in the field. And we've been doing this for 5 years, and I'll be darned, we cannot tell these four species apart in the field. They are morphologically cryptic. And that's really a problem, because if we want to do downstream studies of, ecological differences or reproductive isolation or phenologic, anything that we want to do downstream is made that much harder because we can't go out in the field and know what we're looking at. So that is a problem, but we have a little bit of a workaround in that we use the genome data to create these barcode markers that do not need to be sequenced. They simply, we found regions in the genomes that if we amplify the different species, they give us different size amplicons. And first we separate medium and divinum, and then once you know what it does at this first LG9S1 primer, then you go to the subsequent primer, and you can see that's the first step, the smaller and the bigger, and then separate the species. So this really works quite well. Not perfect. It would be nice to go out and know what we were looking at. But we've now done a lot more sampling, and these barcode markers allow us to resolve what are the geographic ranges of these species. And interest, and particularly of interest, is do they occur together? Could they interbreed? It's one thing to see that they don't interbreed if they're always allopatric, but we need to know if they occur together within peatlands and could interbreed if they're compatible. So this is some sampling that we've done up and down. You can see, for example, that it turns out that only Sphagnum divinum occurs in Western North America as opposed to Eastern. Sphagnum magni only occurs in the southeastern coastal plain, all the way down to central Florida, whereas the other three species are boreal, or boreal cold temperate in their distribution. So it really allowed us to understand the geography of these species. And then what we did is we actually went back and we sampled a number of populations more intensively. And this is a result of that. And these are populations. Each column is a population. And the columns show, this column is pure Sphagnum divinum. This column is part Sphagnum divinum and part Sphagnum diabolicum. And the width of the column is the sample size. So we looked at this. What we see is that more often than not, by far, these species are occurring sympatrically. And in many cases, all three northern species are occurring in sympatry. Now, peatlands are big places. That doesn't necessarily mean that they're sympatric at a scale that matters in terms of mating. Remember, they rely on water for sperm dispersal. So we actually went back and mapped the individual samples as we collected them, not having any clue what they were. But then we used the barcodes to identify them, and then we were able to go back because we georeferenced each collection. And we found, for example, that Sphagnum divinum, like the Norwegians suggested, does often occur in the surrounding forest, but it also can occur around the water. And we have any number of places where three species occur in close proximity to one another. And this is just one example map. We see this over and over again at site after site. So they are occurring sympatrically at a scale that matters in terms of reproduction. Now, so we took these sites that are sympatric. These are based on just sympatric sites. And we used structure to look and ask whether there's genetic admixture. And whether, if there is genetic admixture, is it at sites where they occur together? So it's suggesting that there's current gene flow. And we found divinum and medium are absolutely distinct, but magni and diabolicum, these two closely related things, really don't differ much at all, except that magni has an additional genotype group that's present in higher frequency than it is in diabolicum. And if we do all sorts of pairwise comparisons, we see what appears to be complete reproductive isolation. Now, I want to acknowledge that you can see something like this, and there still could be some level of introgression. So this is not the final word. We're actually working with this data set right now to more explicitly test for introgression in these sites. But I want to focus in the last part of my talk on Sphagnum magni and Sphagnum diabolicum. Remember, this is the phylogenetic relationships, and recall these three species are boreal, cold temperate. Magni is semi-subtropical to cold, warm temperate. And this tends to be in the open peatlands, and this is in the open peatlands and in the surrounding forests. So there's a little bit of ecological differentiation, and we sampled up and down the East Coast, and our sample sizes have increased. And this is what they look like. These are you already saw this in a separate picture, but this is a typical boreal peatland. Sphagnum magni grows in a place that no self-respecting sphagnum would ever grow. I mean, and I'm used to doing a lot of sphagnum field work, and I'm always bear conscious. In Alaska, it's brown bears, it's black bears. I've never had to worry about gigantic alligators when I'm collecting, but those are the habitats the Sphagnum magni grows in. And what we found, again, looking for genetic admixture and lining up our sites along a latitudinal gradient, at the southern end of the spectrum, it's pure magni. At the northern end of the spectrum, it's pure diabolicum. But from North Carolina to New Jersey, almost all the plants are genetically admixed. So there's a region which is basically where the two species ranges overlap. They're genetically admixed. So interested in those, and we wanted to know, are we seeing a signal of incomplete lineage sorting? It's just ancestral polymorphism, or are we seeing a signal of introgression? And so Brian, my former student, compared models, basically coalescent simulations to compare the best model to explain the patterns that we saw. And he tested models where there's been continuous gene flow since speciation, gene flow at the beginning, then isolation, and then secondary contact, or just gene flow briefly subsequent to speciation, or secondary contact only. And this by far was the most highly supported model. So we do think that there's introgression going on between these species. We were able, because we have genome sequences for so many of these, we were able to look at a little bit more detail of what are the genetic factors that distinguish the two species. And so we did FST scans across all 19 chromosomes, all 19 autosomes, and identified regions of the genome that were more distinct than you would expect from background FST values. And as has been found in some other organisms, those high FST, those high regions of differentiation with a lot of fixed differences occur in these discrete genomic islands that are characterized by high FST, low genetic diversity. And so these islands we were kind of excited about. We knew from that same study that divergence between the two species is heterogeneous across chromosomes. So chromosomes 1, 11, and 16, obviously 1, 11, and 16, account for 40% of all of those windows, all of those outlier windows that we identified. And those outlier windows contain over 500 genes, or 91%. They weren't just scattered around the genome. 91% of all the fixed differences between these two species occurred in these discrete genomic windows. And 10% of the fixed differences were within genes. So there were 645 that result in missense mutations that are good targets for learning something functional about what's going on here. And the pattern of variation, FST, DXY, and linkage disequilibrium, et cetera, again, I won't go into the theory behind all this, but fits a picture of predicted selective sweeps accounting for this. And we found through McDonald-Kreitman test that there were 10 genes that showed evidence of adaptive evolution among these things. And 7 of the 10 genes are actually physically in these genomic islands. So here's a list of the genes. I'm not a functional genomicist, so this basically I'm happy or sorry to say is where I stop. I'm the field guide. But here are 10 genes that I think are good candidates for underlying ecological speciation in this group, and that may be good candidates to start for ongoing research on what they're doing and how they impact this northern cold adapted species and the southern warm adapted species differently. So I've been going a mile a minute. I hope you haven't that. I did it purposely because I usually talk for 2 hours in a 45-minute slot. So I want to just leave you with these messages. I want to remind you that we chose Sphagnum magellanicum as one of our target species for the genome work because it had this incredibly widespread geographic distribution. In North America alone, it extends from subarctic Canada all the way to alligator-ridden swamps in Florida. And we thought, and part of our dimensions of biodiversity research was to study differential adaptation along that latitudinal climb. So we went into it, and we realized early on that Sphagnum magellanicum isn't one species, it's 7 species, at least 7 species. I will note that we don't really have samples from Africa or Western South Asia. So at least 7 species. I think we seem to have caught speciation in the act, especially in the Diabolicum and Magnum. They're incompletely isolated reproductively. They're much less genetically divergent than the other pairs of species. etc. So it turns out that, well, and I will say, well, it turns out that the group is not only a great group for pursuing climate adaptation, because it does include northern subarctic species and subtropical species. It's just not within one species. But what we've learned is it's also a tremendous system for studying speciation. I would argue one of the best in bryophytes. So we're very excited to move forward. And so far, we've really focused on divergence. That is, what are the fixed differences? Where are they in the genome? What genes are they part of, et cetera. But we're also very much interested in this issue of introgression, which we've started to get at. And there would be lots of, and I will say that if NSF had been more cooperative last year, we would be doing that right now, but maybe next year, to really pursue what's going on with introgression and doing common garden QTL kinds of based studies with this. So I'll leave you with, obviously I'm a small cog in the program here. We've had good financial support from the National Science Foundation and from the Department of Energy, both JGI, which is Department of Energy, but also separate Department of Energy grants for the microbial work that I didn't really have a chance to talk about. At Duke, one of my graduate students, Karn, who just finished, sadly, my last graduate student, I think, before retirement, and several postdocs, as well as Blanca, who works in the herbarium. We all have worked together on this. Brian and Dave at Oak Ridge National Laboratory, Hudson Institute of Biotechnology did the genome sequencing for JGI and also our Dimensions supported genome sequencing. Jeremy oversees that. Adam is the computational biologist, without whom I would have just had my eyes rolling with the data. And then also a couple of field colleagues who have played a big part. Sean Robinson, who I was in the field with yesterday, collecting these things at a site north of Syracuse, and Scott Schutte, who took me around to peatlands all over Pennsylvania as part of that sympatry study to see what grows where. And with that, I will finish. I don't know how to turn on the lights. So I might have a little bit of time for any questions.
You look for microscopic differences.
Well, we published a paper a couple of years ago with very detailed microscopic studies, and the conclusion was simple: we can't tell them apart. Really thorough. Every character I could look at. They really are cryptic, morphologically cryptic. I haven't given up. We're still working on this to learn how to tell them apart, but they're pretty darn similar.
To follow that question, how do you write a new species description when all you have are some gels, these products?
Well, it's not the first time I've been faced with that, and I've, to fulfill the letter of the law, in some years past, I said, for the species diagnosis, a mutation of an A to a T at site 327. In this case, I had to do the same thing. These two species just came out a couple of months ago, or 6 months ago. And, we had say, like Sphagnum magni is often green, much less commonly red. And we just said Sphagnum magni, often green, much less commonly red. Completely useless for identifying the species. We were simply fulfilling the nomenclatural legal obligation. It's a problem. And there are people that hate our guts for describing these things. And I had many discussions with people, both at Duke and outside, and we had dissenting views even within Duke, within our group. These shouldn't be described as separate species. But my argument is they are very important units of biodiversity, climate-wise, niche-wise, evolutionarily. They have to be named. If they can't be distinguished morphologically, well, I apologize, but there's more to taxonomy than just identifying specimens. There's cataloging biodiversity and important biological diversity. That's my spiel. There was enough, yeah. Wait a minute, I need you. Still the genes.
You found any genes that regulate?
Oh, no, heavens no. I wish. You know, Sphagnum is a great model for genomic kinds of things. But one of the biggest problems is that no one, including us, has learned how to grow sphagnum from a spore to sexual maturity and do a cross. So we are really limited so far in what we can do genetically.
Subjected under experiments as levels for game, we would also fall apart into.
Yeah. Well, we've looked at other complexes of species, and the answer is sometimes yes, sometimes no. There's plenty of species that are just clearly distinct, and when you look at them, relative to, widespread sampling and also relatively related things, it doesn't turn out that they're part of complexes. But there's another complex that's not that far from this in the same subgenus that I'm very excited to study. That's really had no molecular work at all. And it also has one species that's subarctic and another one that's subtropical. It's got all the attributes that I'd be looking for an interesting system, but not all of them. Which one? The Sphagnum imbricatum group, Aphany, Austinii, Styrii, and imbricatum.
There's a chat question.
I don't understand a question. I want to ask you.
I don't think there's been, there hasn't been as much experimental. There's been a lot of descriptive studies, where they grow relative to pH, for example, or whatever, but not nearly enough experimental studies really assessing how they're impacted by, if that's your question. So there's virtually nothing known. A lot of few more lifetimes of research on this group.
Not necessarily your diagnosis, your description, but are you including some of those information?
In the ecological, you mean in the nomenclatural descriptions of the species? Well, we didn't know them at the time. We knew the molecular phylogenetics. And we did say something, I guess, about niche differences, but the niche differences are subtle. There's a lot of overlap. They're not clear niche differences. As you can see from that one map, they can all of them grow together. Oh, I don't know whether I want to. Oh, then now my son. and he's a foodie. You guys, jeez.
This has been a production of Cornell University, on the web at cornell.edu.